Thank you very much, Albert. First of all, I would like to thank Rilem for organizing this workshop, and also Dr. Esperanza Menendez and Professor Albert de la Fuente for organizing this very nice meeting. The topic of my talk will be the biological concrete, and within this very large title, new developments for living facades. Just a brief outline of my presentation. First, I would try to speak a little bit on bio-based construction materials, green facades and living walls, and why it's interesting for our cities to develop this. And later on, I will talk on the UPC approach, what we have been doing for these last 8 years at our research groups, which is the bioreceptivity optimization of cementitious materials. And I will focus on the laboratory and the natural colonization experiments that we've been doing, and now the upscaling and industrial validation project that we are doing so far. And I will end with very little on the future challenge that we are facing in this project. But before doing that, this project started some years ago, several years ago, in a very different topic. Professor Antonio Aguado and Professor Luis Aguillo started to think on the biological degradation of concrete structures, specifically in concrete dams, and trying to identify or to focus if the biological colonization of these structures can give us some understanding on their degradation and internal processes. But moving from that topic, they decided or tried to develop a new concept of concrete, a living wall of concrete that is made with this biological concrete. And this was the PhD thesis of Sandra Masso, which most part of her work I'm presenting today. This work, I've been part of the research project, and also I was the co-supervisor of the PhD thesis of Sandra, but it's mainly her research work during the last 5 years. So there are many different topics at this moment related to the bio-based concrete products. One of the most interesting, and there's many different research papers at this moment, is the bacteria-based self-healing concrete. But we also have the development of natural fiber concrete, bioconcrete, biogrowths, and of course the development of green roofs and facades. There is an increasing interest on these kind of materials, but I will focus on this one, the development of green roofs and facades. And I would like to show you some examples that what is being doing up to date in this topic. These are some examples. This is in Singapore. This is a green roof, also in California. These are not green concrete. These are structures in which we settle in the upper part of our structures a very complex media in order to have this green growth. Of course, we are increasing the load of our structure, and we are having a very high maintenance cost for these materials. We also have, this is a very famous green wall that you can see in Madrid, in CaixaForum Museum. This totally located in the wall of a closet building, and it's a different approach in which we have the plants located in the wall of our building, but again we have very strong maintenance costs, and also most of the times there is a problem that we are not taking into account. Most of the plants and the living plants that we are having in these walls are not specifically from our environments or our countries. So there is another topic related in how they will survive in this environment. So we have again a very high cost on the renovation of these plants. These are other examples that are very increasing across the world, in Provenza, in Italy, and in the Canary Islands in Spain. So, but why we have to focus, or why there is interest in having these green walls in our cities? This is some data on the percentage of green space covered within the city. So we can see here these red points all across the world, and it's increasing the loss of green areas in our cities. And it's a very specific concern of the public administrations. And some examples on this interest of our administrations in how to increase the green areas within our cities. This is an example, it's a project in Madrid in order to how to increase the green areas. This is part of this project, and how to increase, for example, taking the chance of walls that are not used for any other application. But also, this is another promotion from the Barcelona City Council. They are starting a very big project. At this moment, it's just a collection of ideas, but they will give helps. I think it's up to 100,000 euros per project in order to increase the green areas in our cities. So there is a very high concern in our cities for this kind of solutions. But in fact, we have biological growth in our concrete structures. It's naturally it's everywhere. This is an example of a hydroelectric canal that is located in the north part of Catalonia, and you can see in the walls of the canal very large growth of algae. So it's very normal. What we have, or what we need in order to have this biological growth, is the appropriate environmental conditions, the presence of organisms, and also the bioreceptivity of the material. In the early 1995, Gilead defined the bioreceptivity as the aptitude of a material to be colonized by one or several groups of living organisms without necessarily undergoing any biodeterioration. The fact is, we have been dealing this colonization as a problem in concrete, because it causes many deteriorations in our structures. For example, in water canals or in residue tubes. But we can think also in this colonization as a benefit for our structures. If we can enhance this colonization and we can promote the colonization of our structures, it can allow us to obtain greener structures. So we started this project in order to understand and to think how we can enlarge and how we can promote this colonization of organisms in our structures. Our first attempt was to reduce the pH of cement. We tried to use different admixtures or different cements, but the fact is that the normal pH of cement, that is around 13, it's very difficult to reduce it to a level that can allow us to have biological colonization in a real time. So what we decided is to use magnesium phosphate cements, that are materials that are actually being used for dental implants, that are biocompatible and have a pH that is close to 7. So this is the first paper in which we presented some of the results on this development. What we started were a large experimental campaign on different compositions to find an appropriate phosphate magnesium ratio, and also to evaluate all the properties of this new material to be used in the construction industry, not in the biological or the dental industry. This moment is also a very interesting material. Since the last 5 years, there are a lot of publications in the use of magnesium phosphate cement. So these are some of the results on the evaluation of the mechanical properties and also on the temperature development during the hydration of this kind of materials. But there is not only the reduction of the pH of the material to allow the biological colonization, we also need to have an adequate porosity and adequate roughness in order to allow these biological living organisms to colonize the surface. So we made an approach in order to obtain a controlled porosity material with a large porosity that we can have enough water inside of our material to promote the biological growth, but also we have a very good roughness in order to the organisms to colonize the surface. So following this approach, in order to estimate the amount of cement paste that we need in order to have this porosity in our material. We obtain the dosages that we are actually using, and we evaluate this bunch of mixes in order to determine which was the roughness and the bioreceptivity of these materials. These are some of the results that were done in collaboration with Ghent University with Professor Nel Develli, in which we evaluate different roughness of both OPC and MPC materials, in which we were varying the characteristics of the cement samples, varying the water cement ratio and the final porosity of the material. You can see that the roughness is very similar in these kind of materials, although we can see the final surface is very similar. At the end, the samples that were selected to be done on the real scale test were the larger roughness. So, after the development and the study on the dosages of this material, we prepared a very extensive laboratory and natural colonization experiments. We settled different samples both in natural exposure conditions and outdoor conditions, and also in laboratory to make the accelerated algae fouling test developed by De Muynck et al. At Ghent University. So, this is a very well-known experiment in which we have a cultural algae that is being pumped to the upper part of this steel frame, in which we have set different samples. So we are spraying the surface of our samples continuously with this algae culture, and we have some lamps in order to simulate the day-night timelines. And this process is repeatedly across the day in order to accelerate the colonization of our samples. And we made colorimetric measurements, scanning images, and photosynthetic activity in order to control the growth of the colonization in our samples. And also we made CO2 absorption at the final of the test in order to evaluate in which of the materials you have the largest biological colonization. So these are part of the results in order to how do we measure the colonization by controlling the wavelength according to the presence of chlorophyll. We use it green algae, so we can control very easily this increase in the absorption of the green light. So these are the rough results, but these are the treated results in order to see how the fouling intensity, the area of fouling changed with the duration of the test. So we have here, these are the OPC samples and the MPC samples. And also, at the end of the test, the dry weight and the CO2. What we have seen is that the MPC samples were colonized earlier than the MPC samples, and in a larger extent that we've seen for the MPC samples. So the presence of this different pH material and also the roughness of the materials allowed us to have a larger colonization in our samples. This is an image of the whole exposure that we made with different roughness, controlling the colonization of the different samples. Further, we move on the natural colonization experiments. We settled 3 different experiments. One was located in the Montseny Natural Park in Barcelona. The other one was located just here in the UPC, and another one at the Magnell Laboratory in Ghent. Unfortunately, we had a very good results in the lab. We have a very high and soon colonization of our samples, but after 1 year of exposure to the natural conditions, we almost did not see any biological colonization. That was in Spain. Again, we see a little growth of algae on the surface of the samples, but in fact, the colonization, the biological colonization of the samples was not very significant, and there were not significant differences between MPC and OPC samples. We did have very small differences. But the point is that we were doing something that is very extreme, since we are not doing any kind of interaction with our samples. So we are expecting direct natural colonization of our samples. In the nature, it will take a long time to do that. So the point and the main challenge in that topic is how to do this biological growth in a real industrial time. So at this moment, we are working in this upscaling and industrial validation. We are within a project with BASF, Manini Prefabbricati, and Escofet. Manini Prefabbricati is a large Italian company which is devoted to the fabrication of prefabricated panels and other prefabricated construction elements. Escofet is a Catalan company which is devoted to the construction of urban furniture made with precast concrete also, and BASF is very well known. And at this moment what we are doing is the development in an industrial scale of this concept, which is the biological wall or the green wall, in which we have a structural layer which will be connected to the actual structure, a waterproof layer that avoids presence of water or the increasing of water from the biological layer to the structural layer, and we have a biological concrete layer in which we want to have this biological colonization of our structure. So in this project at this moment, these are some of the results that I will show on this overview. We are trying to deal with the increase of the water retention capacity of our elements, since one of the main issues that will control the biological colonization in our samples will be the presence of enough water. Also, to optimize the composition of our materials in order to use them in a real industrial scale, because it's not the same to fabricate a mortar sample in a laboratory than to go to a real industrial plant and trying to develop a panel element with our dosage, and also to evaluate the real scale production of these elements. In order to increase the water retention capacity, this morning we have a very nice talk on the use of superabsorbent polymers. We are also evaluating the use of these superabsorbent polymers in order to allow to have a water retention reservoir inside our material. So all the presence of water that can increase from the exterior, it's retained at the largest time possible to allow the biological colonization and the biological growth in the surface. We evaluate different sets of samples with percentages, varying percentages of superabsorbent polymers in order to evaluate the weight increase, and also which is the loss of this water with very simple tests. We also made an optimization of the compositions in order to use non-laboratory products. We evaluate the use of fly ash in order to reduce the content of magnesium phosphate cement, and also the use of an industrial byproduct of a magnesium byproduct in order to also reduce the cost of this material. But the mechanical results compared to the reference of the OPC were not so good, so we have to rely on the use of normal dosages. The other thing that we changed in many of the literature, you can see the use of ammonium phosphate in order to make the mix of magnesium phosphates. But the fact is, when using ammonium phosphate as the source of phosphate, you will have ammonium release in your medium, so it's not very acceptable for an industrial company to have ammonium to be released. So at this moment we are using another source of phosphates in order to meet the security requirements. And at the end, these are some of the photographs of the real scale production that we are facing at this moment. These are some real elements produced some months ago here in Spain. and these were produced some weeks ago in Italy. We are also dealing with the connection of these two layers, since we have to ensure that the biological layer, although it's not a structural layer, has to be connected, and we have to ensure that there is no cracking of the connection of these two layers. So we were evaluating these steel formworks in order to produce our elements, and these are the aspects of the final panels that are at this moment being to test. And this is just 24 hours after the production of the elements. We are having a compression resistance of the biological layer around 8 to 10 MPAs, but there is a very good adherence between both of the elements. The movement of all the panels was really, really okay. So, just coming to the end, the future challenge that we are facing at this moment, these are two different steel formworks, steel frames that we have at the factory in order to deal with this very big problem. That is that we need real colonization in an industrial acceptable time. We cannot wait 10 years in order to have a biological colonization in the surface of our product. So at this moment, what we are evaluating is how we can increase this colonization after the element goes out from the precast plant. These are two different options in order to make a first settlement of these, or first colonization. We will use algae as a precursor for this biological colonization, and to evaluate how this can allow to a larger biological colonization when the elements goes out to the real environment. We also have a very interesting challenge, which is how we can use superplasticizers that are normally used for Portland cement with this new magnesium phosphate concrete, because there are several problems on the dosage and to the workability, and we cannot enlarge and increase the use of water in order to have a good workability in our material. So, thank you very much for your attention, and any question is welcome.